This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 361, recorded on October 30th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. We are back in studio after 3 episodes of being on the road, and I'm really happy to be here. And with me is Dickson Despommier.
Hello, Vincent.
How have you been, Dickson?
Very well, thank you for asking.
I don't really care. I'm supposed to be nice to you.
You're going to get lots of mail. Stop. I guess you just like to get mail. It doesn't matter what kind.
Today, it's a beautiful day.
It's gorgeous.
15 Celsius.
Superb.
The sun is out. Just a few clouds in the sky.
It's amazing.
And Saturday should be nice for the trick-or-treaters.
That's right. And we even got some nice healing rain on Wednesday.
That's right. Also joining us today from somewhere in Florida, Rich Condit.
Tampa, hello. How is everybody?
Good. We missed you.
Yeah, I missed you too. This is it's good to be back together.
Tampa is not north. What do I usually say?
Tampa's kind of central Florida.
Central Florida, whereas
except it's not really central because it's over on the Gulf on the Gulf Coast. But it's in terms of latitude, it's central.
Yeah, so Gainesville's north central, right?
Yeah, that's correct.
Inland.
How's the weather in Tampa?
It's gorgeous. It's 82 degrees with basically few puffy clouds. That's 27.8 Celsius and about 55% humidity. We're finally out of the lousy weather, and it's gorgeous. It's beautiful.
It could, someday, Rich, I might move to Florida.
Really?
Well, you could do worse.
Because there's no state tax there, and I really don't want to pay state taxes on my bloody retirement stuff, right? So,
but isn't Jeb Bush still the governor?
Yeah. So my family.
No, he's gone.
My family.
He is gone. Oh, good.
My family, in particular, my daughter, who is very liberal, said, "If you move to Florida, I will not visit you." Thank you. Very nice.
Yes, this is the land of hanging chads. So,
but
we
you
have our political issues.
You have to balance things. No state tax can. And you, Dickson, should not expire in New Jersey. You're crazy too.
Oh, I tell you.
But you are. You are.
Oh. I'm not planning on it.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
Welcome back.
Yeah. It's been a few weeks.
3 weeks. I'm sorry. I had a lot of traveling to do and
no, absolutely fine. Yeah.
So that's done for a while.
I've just been kicking around on Friday afternoon. Hey, what I'm going to do?
I actually don't have any more for the rest of 2015. No more mobile TWiVs.
Wow. And I see on the schedule we're about to run out of canned episodes. So,
I have one
things are getting tight.
Where I spoke to some PhD students in Australia. That would be 2 years ago, and it doesn't go out of date. But someone had once requested some TWiVs with PhD students, so I might put that up. It's kind of interesting.
Sure.
I'm not sure where, but yeah, we will have some where we either have to adjust the schedule or you will have to do a Vince-less TWiV.
Oh boy,
it's been done before.
It's fine.
It's true. We did it.
Yeah, I will just. You guys can all record locally and send me the files like you did last time. It's fine.
Yeah.
A Vinny-less TWiV, I should say. I don't really like Vince. All right, this episode is sponsored. Bless you, Dickson.
Sorry.
This episode is sponsored by ASM Education. The ASM Education faculty programs and the ASM Leaders Inspiring Networks and Knowledge program, known as LINK, I have to make acronyms, is now offering a 4-part webinar series for both educators and researchers. The series is called ASM MOOCs, where the M could be micro, right? Micro MOOCs. ASM's version of massively open online courses. And the series theme is successful integration of research and teaching. I know about that, Dickson, don't I?
You do.
All right, you don't have to say yes.
No, but I agree.
I'm just kidding. Online, I'm trying to pull you into the conversation. Online registration is open for the following webinars, and one is Conducting Authentic and Inquiry-Based Research with Lee Hughes from the University of North Texas. That'll be October 21st, and that's done, so forget it. It's over.
We should probably update the ad.
This ad I got a while ago, and I was not recording locally. But this one is publishing Ethics and Responsible Conduct of Research with Roseanne Sandry Golden from the University of California at Irvine. That'll be November 18th. And you can find out more or register. Visit facultyprograms.org. Now, I'm going to have a special link in the show notes. And this is a custom link which tells people at ASM when you click on it that it came through TWiV. We'd like you to use that. Not that we get any money for it, so that they know how powerful the TWiV network is.
Yes.
And our listeners. By the way, guys, did you know? Remember, I told you, our listeners, a few weeks ago to vote for the Susan Nassif, the comic author.
Right.
She was on I forgot, an in vitro gen hero. She won.
Did she?
Excellent.
That TWiV Army. And
TWiV.
somehow she got a breakdown of where the votes came from, and the majority came from TWiV.
That's not just a bump. That's a mountain.
TWiV Army.
So
This is power.
TWiV
We have
Nation.
power, we're
TWiV
not-
Nation.
not just a bunch of mooks.
Hey,
Nope.
there you
A
go.
bunch of mooks.
Not a bunch of viroids.
Yes.
So listen, even though this one webinar is past date, are they archived so that somebody can go back and listen to it?
That's
No,
a good idea.
Good idea. I'm going to click the link
I'm
right
going
now.
to the link right now to see.
Well, there are a couple that are done. The
If
only
it's
one
not,
is
it'd
that
be a missing link.
It's showing registration closed
for
for the
Oh,
20
for the-
for all the earlier ones.
yeah. The, November 18th is still open. You can do that. It doesn't say whether there's archived. I would think so. If you're registering, you have to pay. ASM members $25 per webinar. I bet you get something, but you could ask them. There's an email contact.
You wouldn't be able to ask the speaker any questions, probably, but you could.
You could still do it. Although if registration's closed, that means there's nothing there to look at anymore, I would
Right.
guess, so maybe not.
I'm sure that the publishing ethics webinar will be quite something.
That should be good.
Yeah.
Roz is the editor-in-chief of Journal of Virology, so she's up to her neck in this stuff.
Yep.
So.
All right, we have a couple of follow-ups. One from Akira, who was our guest on the last TWiV out at Ann Arbor. Thanks again for having me on TWiV last Friday. That was great fun indeed, and I am relieved to find that I did not make too many problematic comments in the show. Just a couple of minor points. I saw on the TWiV page that my family name is spelled Ono, O-H-N-O, like Apollo Ono. He's the speed skater, right?
Yep.
My family name is actually Ono without an H. If you get a
like
chance,
Yoko.
Yes. If you get a chance, could you change it? And I did change it, but I thought you would like to hear the rest of this. It is not a day and night difference, but it is a matter of size. When written in Chinese character, kanji, Ono with the H means large field, whereas Ono without the H means small field. A small field would be fine for me. Akira is a very funny guy. You should listen to that TWiV because he has a good sense of humor. Another thing is my misspeaking on dinosaurs in Japan. As Kathy let me realize, I described the lack of dinosaur fossil discovery in Japan in the present tense. What I meant was that a dinosaur had not been found in Japan when I was a young kid. Books for Kids explained that since Japan was under the sea, it would be difficult to find fossils of dinosaurs, which were land animals. No longer. There have been dino fossil discoveries in several places in Japan in the past few decades, and nowadays kids in Japan can have a dream of digging out dinosaur fossils without going to foreign countries. I suppose my remark on this can be viewed as just a grammatical error, and probably nobody would care, but if fact-checking correction is important even for non-viral stuff, please let me know. I would be happy to look into the history on Japanese dinosaur digging. It's funny, when we were having the discussion on TWiV, I was going to ask him, are you sure there aren't fossils after you were a kid? But, I didn't want to take it down a non-viral track. I never do that.
No.
So how old is Japan in terms of its life above the ocean? No, seriously, because, if you knew that, you could have automatically said whether or not fossils are possible or not.
Well, if they did find them, it's either undersea dinosaurs. Were there undersea dinosaurs? There were, right?
Yeah.
There were
Godzilla
sort of
comes
semi-
out.
Godzilla, that's right. That's Godzilla.
Godzilla, right.
No, mosasaurs, those are dinosaurs. They're swimmers. And there were flyers also, so they could have flown to Japan.
Like Mothra.
Like Mothra, exactly. Or the giant turtle, whatever that was called.
It was a giant turtle?
Yeah, it was a giant turtle. Flying turtle, of all things.
A whole sequence of people in rubber suits falling.
Same actor, they had to do quick changes.
Exactly. This was a formative incident in my youth.
That's me too.
Well, there's you can find out on the website when it came up, but I don't want to take the time to do it because this is not This Week in Geology.
No, it isn't.
Although it could be, but we need to get some geologists.
Well, you did have a virus that was dug up from the permafrost, which is kind of a relic.
Yeah.
It is.
And we do rock.
And we do rock. This is exactly right.
Rich Condit, can you take John's, please?
Okay, I'm trying to look up when Japan emerged from the ocean.
You want to do that?
No.
Sorry about that, but
So,
tended to
John writes, "Hello, Vincent and friends. I was listening to episode 357 on my way to work today, and a point came up that caught my attention. At one point, the TWiV gang begins talking about viruses as energy parasites who harbor no metabolic activity. While based on current knowledge, this is essentially true. I do think it is worth noting that there has been a discovery of phages which encode metabolic proteins. Here I've linked a paper, and he gives a link titled Metabolic Reprogramming by Viruses in the Sunlit and Dark Ocean. Long story short, phages in the ocean are able to capture metabolic genes, even photosynthetic metabolic genes. How cool is that? To manipulate host fitness overall, or just directly to shift metabolism in favor of the replicating viruses. This is only an example of how viruses can play with host cell metabolism. However, to me it implies that perhaps metabolically active proteins could even play some role in virion or genomic stability outside of a host cell. That may be a bit of a stretch, but biology is all about surprises. Perhaps this paper or a related one warrants at least a snippet in an upcoming TWiV. I'm writing from Medicine Hat, Alberta, Canada, where today it is a wispy mix of sun and cloud and an oh-so-comfortable 3 degrees Celsius, 94% humidity. Wow, 94% humidity at 3, something like that, and a 7-kilometer southward wind. Keep up the great work. You've managed to create an amazing information-filled podcast that is informative enough for me to enjoy, but not so needlessly bogged down in jargon that my girlfriend makes me turn it off in the car. Long-time listener and first-time writer, John. Okay, great.
I've been to Medicine Hat.
So have I.
Yeah,
Yep.
it's a good place.
How could you forget going to a place called Medicine?
Right. That's exactly. This is a cool paper. We might do it here on TWiM, but I would like to snippet it at some point because it's a cool story for sure. So, phages, Dickson, you asked that in 357, do any viruses encode metabolic proteins? We said no. Do you consider a phage a virus, Dickson? Of course. So, there you go. They do. I'll bet there'll be some eukaryotic ones that...
It's nice to raise the question then.
But for sure, because, we have the giant viruses encode translational components, so... And they may be used to help reprogram the host to favor virus replication.
Sure.
So I'm sure we'll find other things as well.
This is true.
And Alan, can you take the next one?
Yes. Some mook named Alan writes... You all have talked about Kevin Folta's unfortunate stumble into the GMO limelight, so you'll probably be interested in the bizarre turn this story has now taken. And sends a link to a piece in BuzzFeed by Brooke Borrell, science writer.
The graphic is...
yes, the graphic is quite good.
Is this you, Alan?
Who?
Who wrote this letter?
Yes, this is me.
Thank you. Yeah,
that's how they make tomato sauce, by the way.
Right. So the graphic shows a scientist-looking character in a lab coat looking at a giant tomato that's sitting on a mousetrap. So the Kevin Folta is this fellow who's spoken out quite a bit on the safety of genetically modified organisms and has been kind of a voice of reason in that whole screeching discussion. Yeah. And then was hit with basically harassment through Freedom of Information Act, which is a common tactic of anti-science folks, and they forced him to release thousands and thousands of emails and dug in and took things out of context and used this to argue that the guy is in the pocket of Monsanto. So we've talked about that, and this story, things have taken an unfortunate turn. So Brooke Borrell had been in touch with Folta before this whole mess came out, and she walks through what their discussions had gone around, and it turns out that Folta also hosted a podcast under his own name, or blogged under his own name, I guess, and hosted a podcast under a pseudonym, the Vern Blazik Science Power Hour. Now, his argument through this whole unfortunate episode, when the anti-GMO folks were saying, look, he's not who he says he is. He's taking money from Monsanto, and I think we've talked about how yes, that sort of had the appearance of impropriety, but it wasn't all that it was made out to be. So he was simultaneously, while taking a perhaps inadvisable grant from Monsanto, hosting this pseudonymous podcast, and at one point, Vern Blazik actually had Kevin Folta on the show.
Oh, that must have been fun to
And so he interviewed himself on the show about genetically modified plants. And this all, at this point, looks quite bad, because you've got this guy saying, trust me, I am who I say I am, and, he's doing this sock puppet show with a podcast under a pseudonym, and it's really just quite a mess.
Boy, it's unfortunate.
It really is.
I know you had a long Twitter discussion about this, Al.
Yeah. Yeah, I think he really screwed up. I think he needed to be a lot more careful about appearances here. I don't think he did anything wrong in terms of, actual ethics, but I just think if you're going to wade into some discussion that is that acrimonious, you need to be very careful about how your relationships and activities look.
Sure. Which we have said
yes
on TWiV before, and he was not careful enough. But this podcast, did he start that after the Monsanto thing or before?
No, he started that. This all happened before the whole thing blew up.
So, Alan, did you discover this Blazik pseudonym thing?
No, I learned it from this story that I've linked to.
This BuzzFeed story. Yeah.
I see.
By Brooke Borrell.
So this is all Brooke Borrell. Breaking the story, and she does a very good job with it. I think it's a very even-handed explanation. But
the emails that I sent through my private line had nothing to do with national security, okay? Let's just stop there. Come on.
Leave it alone. Email, Dickson?
Yes.
Because it's the same type of thing. The more you say it isn't, the more the people look for links, and then they find them, and then they say, aha, you're lying. And then, of course, the next thing you know, you're history.
Yeah, right. You better watch out, Dickson.
No, I have nothing to hide.
You're a public figure.
I have nothing.
Nothing at all?
To hide.
All right. Now, I want to talk about a story that I learned about when I visited the University of Michigan in Ann Arbor. Now, for those of you who haven't listened to the TWiV, Ann Arbor Department of Microbiology and Immunology was honored by the American Society for Microbiology. It was named a Milestones in Microbiology site, which means it played a historic role in the development of the field. So I was fortunate to be able to go and record a couple of podcasts and partake in the ceremonies, and part of that was an infectious disease physician by the name of Paul Kazanjian, who talked about the first chair of that department, Frederick Novy. It was the first department of bacteriology in the country. And, many of us who are in microbiology departments, they started as bacteriology, bacteria, and then we learned that there's more than just bacteria that you can't see. So they became
I think the department was established in 1888, something like that. Is that right?
Yeah.
So that would be essentially before we knew there were viruses.
Right.
Yes.
And my department here, which used to be bacteriology, was founded in the 50s, and it became microbiology, and now microbiology and immunology. Anyway, Kazanjian, he's also a historian of science. He got a PhD in history to go with his MD, and he gave a talk about Frederick Novy, because, of course, he founded that department, which was being honored. And Novy was born in 1864. He got a Ph.D. in physiological chemistry in 1890 and then an M.D. at the University of Michigan in 1891. He studied bacteriology with Robert Koch in Berlin and Louis Pasteur in Paris. Can you imagine?
Pedigree.
Holy cow. And Kazenjin was talking about it. He had pictures of his lab notebooks, so intricate and detailed. Anyway, at Michigan, he became a professor of bacteriology. In that department he established in 1902. He became dean of the medical school and did a lot for microbiology. That's why one of the reasons why the department was feted. Is that the right word? Feted?
Yes. Yep.
For this. Now, in the audience was his...
F-E-T-E-D,
yes,
not F-E-T-I-D.
That's
yes.
correct. In the audience was his great...
Although, as a bacteriology department...
It is feted, yeah. In the audience was his great-great-granddaughter. And we mentioned that, I think, on TWiV. And I saw her. I said, "Are you a microbiologist?" And she said, "Yes." She works for a small company out there.
Cool.
So it's very cool.
So can I interject just one?
No.
Former chairman of this department here,
Harry Rose?
No, Harry Ginsberg.
Yes.
Is a graduate of University of Michigan.
Yeah, that's right.
He played football for them.
Played football there, yeah.
That's right. So that was a hotbed of education for this subject.
Our department was founded by Harry Rose here.
Yes.
Yep.
I knew him.
Anyway, one of Novy's students was Paul de Kreef. Anybody know that name? Alan's for sure.
I don't.
You don't. He wrote a book called he wrote a book called Microbe Hunters.
Oh, I know the book. Why don't I remember that name?
Okay.
That's right. Yeah,
but he also worked with Sinclair Lewis when Lewis was working on Aerosmith and helped him develop the characters.
Good heavens.
And one of the characters, Max Gottlieb, a fictional medical professor of bacteriology, was based in part on Novy.
Cool.
So Powell talked about all this, which is
circles
really cool.
of interconnectedness.
Now the cool part is that Powell mentioned this. So Novy worked on culturing things. He got really good at learning how to grow various bacteria. There's a Clostridium species named after him. He worked on propagating trypanosomes and leishmania.
How about that?
And spirochetes. And he used to get samples from people because he was so good at this, and he would put them in various animals and culture.
Man for all seasons.
It's a man for all seasons, that's right. Have you ever heard of him, Dickson, in conjunction with parasitological research?
Sorry that I haven't.
Why are you sorry?
Well, because I would have enjoyed knowing more about him. It sounds like he was an interesting man.
Now, he had gotten some samples that were supposed to have spirochetes in them, and he put them in rats, and he got them to grow. And then he ran into a situation where his rats were dying really quickly, and he figured out basically that there was a virus, or he thought it was a virus in there. Now, this is 1910.
Wow. Yeah, so it was a filterable agent.
Called it a filterable virus, which because virus was used at the time for any infectious agent. Okay, but filterable virus. So this is an interesting point. Filterable virus was something that passed through a porcelain filter. Yep. And of course, that was the criteria for showing the tobacco mosaic virus, which was at the end of the 1800s, right? 1899.
Right.
So Novy provided some evidence that this agent that was killing his rats really quickly wasn't the spirochete, but was rather a virus, right? A filterable virus. And he said, well, we need an ultra microscope to see this. He said this in early in 1900s, way before it was developed, right? So then he actually showed that you could pass this filterable virus from one rat to another, and he published a paper in 1910, right?
Did he actually publish anything in 1910?
Yeah, he says he reported in the reports of the Michigan Academy of Science in 1910 that you may not have that journal, right? That a filterable agent rather than a spirochete bacterium or trypanosome was responsible for encephalitis on inoculation of filtered blood into experimental animals. So he worked on this for about 8 years. He showed that you couldn't grow it in the normal ways. There was no cell culture back then, remember?
Right.
So we had to infect rats and pass it from rat to rat. In fact, his laboratory assistants every third day would take blood from a batch of dying rodents and infect new ones.
Wow.
In fact, this is how many viruses were passed in those days from animal to animal.
Their assumption was that they couldn't store it.
That's
that
right.
It needed to be continuously passaged.
Well, also, we didn't have any freezers back then either, right? The freezer wasn't developed until...
Well, that was in Michigan. That's called winter.
Yeah,
Yes.
that's right. Everybody has to do their research on Michigan. So from 1909 to 1918, he did this rat-to-rat passage. Suddenly, in 1918, they lost this filterable virus. It just disappeared. It could no longer infect rats. And
Tell me it became attenuated and it didn't kill us. They didn't know it was there. Could have still been there.
There was a rap about sort of the turmoil that went along with the start of World War I.
That's right.
Involved staff leaving, and so that interrupted the rat passage, and then somehow the tubes that they did have with blood in them that had been stored were somehow misplaced.
Yep.
So the research stopped because of all that.
Oh, gee.
So it stopped. In 1935, he retired, sort of like Dickson.
Right. Except I didn't have his rich history.
Yes, I don't know what he did, but
Fished.
they were cleaning out his old lab or the old space in 1951, and they found tubes which looked like they were his. They were 25 dust-covered tubes. They were sealed with glycerin. They were at room temperature. They had a label on it. It had blood in it, but it was all dried out. So they called up Novy, and he came in and he looked at it, and he said, "Yeah, that's it." He'd been living in Ann Arbor during his retirement. He came out of retirement.
Oh, my.
He still had his lab notes.
Oh, my goodness.
40 volumes.
Yow.
Now, I read this, and I felt bad because I've thrown out about half of my lab notes because I always figure, who the hell would care?
Now you know.
But you see, he was so meticulous.
Wow.
Now,
So-
you see, Vincent, if somebody finds old vials from your lab, there's going to be quite an incident, I suspect.
Oh,
Yeah.
I don't want to find polio. So he came out of retirement, and this is... We'll talk a little bit about the papers. There are two papers that were published in 1953 describing his findings, but the blood was dried. So what they did is they took the tubes, the blood, and the labels and ground the whole things up.
Right.
Ground it all up. Glass, old blood, bits of yellow labels, put in saline to extract it, and they put this in mice, and it killed three-quarters of them. Sorry, rats. Thank you, Dickson. I'm glad you're listening. It killed.
Yeah, I just woke up when you said mice.
It killed three-quarters of the rats in 3 days.
No kidding. What happened to the other quarter?
Well, they survived, but there probably were only a few PFU in there, right? It's amazing that there was any.
Mm-hmm.
It's true.
And then
Oh, now you can find out what kind of virus it is.
he looked at it.
Exactly.
He had someone working on his PhD in this. He got someone to do it.
Yeah.
They looked at it under an electron microscope, and they saw virus particles.
Nice.
So they wrote a couple of papers, Journal of Infectious Diseases, and they were published in 1953,
Good Lord.
the year I was born.
That's the year you became diploid.
It became diploid. That's right. So this probably is a parvovirus. I don't think the disease pathogenesis and the size of the particle, 20 nanometers, consistent with a rat virus called Killam rat virus.
Kill him. That's a good name.
Yeah, Killam.
Yes.
What is the name of that law firm, Dickson?
Dewey
kill him?
How?
Dewey Cheatham and Howe?
Dewey Cheatham and
This
Howe.
could be used as a biological control for rats if it's really good.
Oh, yeah, let's release it, Dickson. Good
Why
idea.
not?
Why don't we send it to Australia?
No. Put it in the New York City subway system.
I've always thought you've talked about your favorite virus names, Vincent, and mine has always been Killam rat virus.
Killam rat.
By the way,
No, mine's
for the
chikungunya. Chikungunya. I had that for dinner last night. It was delicious.
By the way, for the listeners, that's not KILL''EM.
Right.
It's capital KILHAM, named after the guy that
Made rugs?
described it.
So this story attracted national recognition. I found a New York Times article from 1953. I sent it to you guys, the PDF,
Wow.
that describes this. It was front page.
Nice.
He could go back, notes 40 years old, and reconstruct this. Wow. And he died 3 years later at 91 years of age.
Goodness.
Then he could finally retire.
He could
That's
finally retire. And this article, which you should look at if you can get to it, by Powell. What can we learn from this? It argues that scientific curiosity and perseverance, characteristics of the younger Novy that TWiV fictionalized with Lewis and Arrowsmith, do not always diminish with advancing age. Now, they don't always, but they often do, unfortunately.
I think it also teaches us that we really, really ought to have some way of archiving laboratory
notebooks, you mean?
Yeah.
Yeah. Yeah.
I noticed also that the New York Times article starts out with quite a bit of overhype.
Sure.
Yeah. Okay, about how this is a deadly killer virus and all that kind of stuff. It's not till they get considerably further down that they mention that, oh, yeah, it's a rat virus. Okay.
Why do they call it a scientific freak?
So a question here, of course, is...
To sell newspapers.
To bring this study forward, ultimately, what kind of virus is it, and what does its genome look like?
We just told you weren't listening.
Yeah, no, you said it was probably a parvo.
That should tell you all you need to know.
Probably a parvo. You didn't say it wasn't.
Well, they didn't identify it.
Parvo
well, you could still, though, can't you?
Hey, don't fight me, Dickson. You should know.
Can you? I'm not fighting you. I'm asking a question. Can you now do the experiments?
Listen to Rich. He's going to explain
Okay, Rich, go ahead.
One could do the experiments. It wasn't definitively identified, but at 20 nanometers, actually, it could be, I suppose, an astrovirus or something like that. But
why have to guess?
If it is in fact a parvovirus, it's a naked capsid virus with a single-stranded DNA genome. It has just 2 genes. It's one of the smallest viruses around. The human parvovirus, there are a couple of note. One is called B19, causes 5th disease, a rashy disease, also known as slap cheek syndrome. And it's also the same family that our favorite gene therapy vector comes from, adeno-associated virus.
That's right.
And there's a couple of other noteworthy parvoviruses of animals. There's a dog and a cat parvovirus that can be a big problem for pet owners.
By the way, I just got a text from Kathy Spindler, who is at the Air and Space Museum in outside of Washington, right near Dulles. Yep. She sent a picture of the Hubble telescope model. Now, I thought she was supposed to be working. Well, it's Friday afternoon. I guess they're done, Rich, right?
Yeah, I guess so.
Well, the Aerospace Museum is in the heart of Washington,
D.C.
that's
that, right. There's also one out by Dulles.
They have 2 sites. They have their the main museum is right down on the mall in D.C., and then the Dulles facility is sort of like a working. It's like a workshop that you can tour as a museum.
I didn't know it was there.
They do a lot of restorations.
The one near Dulles is fabulous, Dickson. They have They have a Blackbird, that airplane.
Oh, yes.
They have a shuttle, a complete shuttle.
We have a Blackbird right here in New York.
They have a B-29.
That's nice. I would like to.
And they have tons of engines and of all kinds, hundreds. I mean, if you were really into this, man, you could spend hours there. Yeah. Anyway,
so
yes, Dickson.
Can we please come back to this virus for a moment?
Yes.
Has anybody characterized this virus the way you've characterized all the other viruses?
This particular one
this particular one.
I don't know, Dickson.
You don't know?
I don't know. I mean, certainly the Killam rat parvovirus has been characterized extensively, but whether this is that, I don't know.
Exactly. So you still have some of that left?
Well, it may have been published, and I didn't. I just looked at these original articles, Dickson.
Somebody should search for some old brown vial.
Exactly.
1953 now.
You got it. That's right.
You know, I got a collection of polio strains from a fellow who retired when I first came here. He was out at Berkeley. And a lot of those vials were from the 40s and 50s. But they were labeled. I knew exactly what they were. I wanted to just point out a couple of things in his paper. Journal of Infectious Diseases, 1953. The rat virus is the title, which I think is awesome. The rat virus. Fred Novy, Perkins, Chambers, and the last author is Paul TWiV, who didn't really have a long career in research, but mostly wrote. He calls, so in this paper, he basically shows that it passes through a filter, and he calls it a filter passer. Isn't it true? The blood of the infected rats was clearly free of bacteria, and this fact led to the belief that a filter passer was present. I just love that,
It's a football term.
Is that right? So he goes through a whole bunch of experiments to show that it passes through filter, how much you need to kill rats. You don't need a lot. You can dilute it extensively and just kill them, whether you get any immunity to it, and so forth. And then he decides that this originally came from the vendor who sold the rats. He got them from various places in the South, and he said there often were dead rats, so probably, he says, looking backwards, it's perfectly clear that the virus was introduced into the lab as an apparent infection. It was probably propagated by lice and other ectoparasites as a relatively mild infection. Maybe, right, Dickson? Maybe. I don't know. Now, the part I want to tell you about is here. They tried to purify it or pellet it by centrifugation. So they had a Danish centrifuge with a maximum speed of 8,000 RPM, which is not a lot for viruses. You need at least 30 to 40,000 RPM, right? And so they centrifuged it for 3, 6, or 12 hours. During this operation, the top of the centrifuge was removed in order to avoid overheating. And they lowered the room temperature to 10 to 15 degrees Celsius to keep it cool. Now, I can tell you that if you tried to leave the top, well, you can't on an Ultra. It won't go. But, you can't. It's aerosolizing. You shouldn't do this. But, back then.
Different age.
It was not pelleted by 8,000 RPM for 12 hours. That's what I wanted to point out. I just thought that was so cool.
And their filtration experiments were pretty involved too.
Very much.
They didn't just put it through a ceramic filter. They did that, and then they went through successive other materials for the filter, a collodion sac, and then they filtered it through. They took the ceramic filter and they impregnated it with agar. They do this. They've got this whole procedure for how to impregnate the thing with agar, and they filtered through that, and all trying to get at, I guess, the size.
Right. Yeah. That's very cool.
Dickson, in one of these retrospective articles we have, this is the closest that I have to answering your question about what it is. Okay. That Novy described the following properties of the virus: highly stable, resistant to extremes of temperature and pH, small size of 20 nanometers, transmitted through the fecal-oral route, producing symptoms of encephalitis in rats. Each of these features is consistent with a type of parvovirus called HER1, or Killam rat virus, first described in 1970. Okay, so that's as close as we get.
How many other parvoviruses are there of rats?
Oh, I don't know.
Why do you want to know how many, Dickson?
Because this could be one of many, that's why.
I'm sure there are hundreds. Now, would you like a number?
Well, no, because if they're all about the same size, then you can't really tell much from looking at the size.
Well, we don't just look at them. We just sequence them nowadays, right, Dickson? I'm going to go. Dickson, tell me something. Where would you go to find out how
Gene
many
Bank.
different...
Gene Bank.
Gene Bank, maybe. But what about the International Committee on the Taxonomy of Viruses?
You could go there.
How about that? Would that work?
I think so.
We go to the current taxonomy release.
Yes.
And it's under families not assigned to an order. They arrange this by order. I don't know why. So we're going to look on the Parvoviridae. Okay, click again to expand.
Good.
Would it be Denso or Parvo? I guess Parvoviridae would be the subfamily. And then we have lots of species, and I don't know which one.
Kill them there.
Well, these are the species names. Let's see. Amdoparvovirus is a carnivore. Nope, so that wouldn't be it. These are birds, galliform. Boca parvoviruses. That sounds like humans. Ungulates. What's an ungulate, Dickson?
It's a hoofed animal with clove hoofs.
Is
that's an herbivore like a cow.
There's a pinniped dependoparvovirus.
That's
a primate. Well, look, a rodent erythroparvovirus.
Erythro.
So there's one type species. So these are only the ones that have been classified. This actually doesn't tell you anything.
No.
Well, if you look at the there's a technical data sheet from Charles River.
Yeah.
That's an animal supplier.
Yes.
It's on rat parvoviruses, and I don't know if these are all different, but they name RPV1, RPV2, RMV, RV. Alias KRV, that's probably Killam rat virus, and H1. So I presume all those are different. So that's 1, 2, 3, 4, 5 that Charles River cares about.
Oh, okay.
And I bet if you looked in wild rats, you'd find even more.
Oh, my goodness.
And if you look on the website of the National Fancy Rat Society, there is an article
from Charles Darwin's day
on rat parvoviruses.
Is that true?
They claim there are 3 groups of parvoviruses, each with a number of strains. But they say the only strain that seems to produce disease in adult rats is the first one described, which is Killam rat virus.
Okay.
Okay,
I
Dickson?
love that.
Yeah, no, fine.
Listening.
I'm here to be the voice of non-virology.
Okay.
Dickson,
I thought I was doing a wonderful job.
Don't worry about my
we need to write down your assignments for projects because you have several, and the next is to exhume this virus and characterize its genome.
And in the meantime, if you take your rats to shows, you should quarantine them afterward.
Yeah.
Dickson, what's going on with that banner over the Hudson?
It looked as though the airplane was heading towards the Hudson River.
There's a banner flight?
It just fell. The banner fell off the airplane. It's falling into the water.
Oops.
Bummer.
It looked, you saw the airplane go by, I saw the banner go by, and then I saw the banner start to go straight down, and I said, oh my God, the airplane is crashing. But apparently it was just the banner.
Just the banner. Well, there's a...
we see things out of our windows that we don't want to tell everybody. We dare not tell the rest of the world what's going
Dickson,
on.
he's waving his hands. I took a picture and a movie of it.
Oh, outstanding.
Yeah. That may make the news. There's a release, usually some kind of release lever for the banner in the plane. So if there's an emergency, the pilot can let it go, but they're not supposed to just do that.
Exactly. You do a lot of flying out.
I didn't see the plane that it came from.
Maybe
it's a Piper
they
Cub
have
of
some
some
kind
sort.
of emergency.
Maybe.
I have a picture. I'll tweet it so that the New York Times can pick it up and put it into their
Geico
thing.
down the drain.
Was it a Geico banner?
I think it was, actually.
Save on your insurance.
Exactly. Oh, yeah, but did you know that?
Are you covered for that? All right.
Did you know that Racaniello is really a Killam virus? They should have named it Norvovirus. Come on.
Right. After the town in...
Right, in Michigan. Or Novy, the Novy rat virus.
All right, let's continue with our podcast.
You bet. Well, this is part of it. Come on, this is the richness of it. This is not just about viruses.
All right, let's talk about Ebola viruses.
A good idea.
Now, don't go away, folks. Don't touch that dial.
That's right.
It's going to be interesting.
This is not your ordinary Ebola virus story.
I know a lot of you are tired of Ebola virus, but this is kind of cool. It's new stuff that we didn't really...
We haven't talked about Ebola for a long time.
We have not.
At least 3 weeks. Right?
That's right.
You should not get tired of this.
Rich Condit, could we have an Ebola sit rep?
I would be glad to provide you with an Ebola sit rep. In the most recent week, there were just 3 cases of Ebola, all from Guinea. Really? There were none in Sierra Leone, and in fact, there has not been a case in Sierra Leone for 6 weeks. And in fact, if they make it to mid-November, they will be declared Ebola-free.
That's amazing.
And Liberia, of course, they are not even reporting on Liberia anymore because they've been clean for a while. Now, there were 2 weeks, the week of October 4th and the week of October 11th, when there were no cases anywhere. But then there were 3 new cases in Guinea on October 18th and 3 new cases on October 25th. And importantly, those 3 new cases in October 25th, they understand where those came from. They were all from contacts, from a close contact of a person who died of the disease. So it's not as if these are popping up out of nowhere. Right. These are all in places where they're tracking these individuals.
So remind us again as to what the incubation period is.
21 days.
Okay.
Is sort of the time that they. Well, no, the incubation period is less. Is 2 to 12
days.
Something like that. But if you the 21-day period is if you are exposed and you don't show up with that's the outside range. If you are exposed and don't show up with disease in 21 days, then you're okay.
Right. 2 to 21 days is the incubation period. So it can take as long as 21 days after infection. Right.
And the 3 new cases are all part of the same household. It's actually kind of tragic. It's a 25-year-old woman who's 7 months pregnant, her 10-year-old son, and 4-year-old daughter.
Now, this I just want to remind people, this is not... sorry, what country was this again?
Guinea.
That was
Guinea.
in Guinea.
So, if you remember, we talked about the vaccine trial, which is being done in Guinea. But apparently, these individuals were either the well, they weren't part of it, right?
And I think it's probably noteworthy as well to point out that during the course of the epidemic, the WHO total is now 28,575 cases with 11,313 deaths.
Goodness.
And also, when we were talking about this back in, I don't know, September or October or maybe earlier, people were making predictions that trajectory that it was on, that by this time of year or later, it was going to be like totally out of control.
Right.
But the public health measures that were put into place got it under control and ultimately worked out.
Yep.
Yeah.
All right, now we have a couple of, one story and a couple of papers about persistence of Ebola virus, which we have touched on before. The idea that after you're declared free of the virus, that is, when they don't find any more RNA by PCR in your blood, the virus apparently can show up or be detected again elsewhere. We talked on TWiV about a physician who had recovered after working in the area, and then months later he had uveitis, infection of the eye, and Ebola virus was actually isolated from his aqueous humor 98 days after onset of infection. Now, the one story for which we don't have a paper yet concerns a Scottish nurse. This is a ProMed Mail story that I'm looking at. She contracted Ebola in Sierra Leone last year, and she was readmitted. So she had recovered. And in fact, I spoke with her initial physician in Glasgow who took care of her on TWiV 341.
That was a great episode.
You like that one?
That was really a nice interview.
People should listen to it. But she recovered. She was not working because she didn't feel up to par, and she had just, I think, gone to talk at a school when she got sick, and apparently she had meningitis consistent with perhaps virus there. Now, the ProMed Mail doesn't have any results of virus isolation or PCR, right? From CSF. So, do you know anything about that?
No, I know nothing about it. So I was curious about that. So, it's not clear why it is or how it is that they finger Ebola in her current illness, except for her history. But I assume they have some lab tests of some sort or another. They also say that although they have her in appropriate isolation, that the chances of transmission are low. So it will be very interesting ultimately to see what really the situation is. I kind of assume that she's not viremic or something like that, but that's only an assumption. So maybe it says
here she is thought not to be contagious. Yeah.
Right. The director of public health said the condition is a complication of a previous infection with Ebola virus. Risk to the public is very low.
So the thought occurs that this is a hemorrhagic fever virus, correct?
It's not the thought. It's known to be a hemorrhagic fever
So
virus.
During the episode that she suffered through, could there not be a second virus or a third virus not related to Ebola that she could have harbored, which, because of the hemorrhagic nature of Ebola, could pass through the blood-brain barrier easier than it could have ordinarily? This could have resulted in that.
Could be, but why would you, if she had Ebola, and we know that it can persist, why would you not take the
No, I would take the simplest. Occam's razor, I know about that.
Occam's razor, you do?
I do. Good. I know all about that. But in addition to that, I knew Occam personally. We used to shave together.
Oh, boy.
Yeah.
Oh, boy.
Yeah, I know.
I don't have any new information at ProMed.
So that's why they should be very careful about what they say about what her cause of illness is. That's all I'm saying.
Well, they didn't say it was caused by Ebola,
yeah, but the conclusion is you almost want to say it's Ebola because she already had it and it's recurring and it could be in the eye and it could be in the brain, but this could be something totally different, but as the result of having experienced the hemorrhagic nature of this.
So here's a story in Medscape. It says a British nurse who contracted Ebola is recovering after the virus caused her to develop meningitis. Doctors have confirmed. That's October 23rd.
Now, is meningitis one of the odd sequelae of Ebola?
Sure.
Okay.
And actually, the ProMed Mail article said the National Health Service Greater Glasgow confirmed that the virus is still present in her body after being left over from the original infection.
So it must be CSF because she was cleared by being negative in the blood, right?
So what all these persistences have in common is that they're in immunologically privileged sites.
Right.
The one in the eyeball, the one in the cerebrus, in the CSF.
Right.
And we're going to touch on some others as well.
So we'll wait for a paper on the nurse. I'm sure we'll see one. Or maybe not, but all right, we'll look for one. Then there were 2 papers that came out in the New England Journal of Medicine concerning sexual transmission of the virus. All right. One, now we did report this on TWiV. This was a gentleman who transmitted apparently infection to his female partner in Liberia in March 2015. So that first paper deals with that. And in this is unfortunately probably behind a paywall, but the introduction summarizes where the virus...
Actually, the 2 New England Journal papers are open access.
Wonderful.
Yes.
Virus may persist for longer periods within immunoprivileged sites. So, for example, in breast milk, vaginal secretions, aqueous humor, as we said, and in semen. Now, that's mostly RNA, and then the virus has been cultured from semen samples collected up to 82 days after onset of disease when there's no longer virus in the blood. So this case was where a woman, March 2015, a woman from Liberia got Ebola virus disease. She died shortly thereafter, and the case investigation didn't reveal any source of infection, but she reported she had unprotected vaginal intercourse with a male Liberian survivor of Ebola virus.
Goodness.
And he had contracted the disease in the previous September, went to the hospital, to the Ebola virus disease treatment unit, eventually recovered and was discharged when he had no more PCR positivity in the blood. So they brought him back and they got some blood and semen samples. And they were unable to culture any virus out of these, but the semen sample at, well, I don't remember the day right here, but one of them tested positive for RNA by PCR, but no virus was pulled out. They have a lovely table here showing you the timeline of all of this. So March 7th, the sexual intercourse, that's 155 days after he had the onset, after he left the hospital. His second blood sample tests Ebola negative. 155 days later, he has sex, and this woman gets infected, right?
Now, I think we mentioned this case,
right?
But this is the actual paper.
That's the paper reporting
This is the peer-reviewed research behind it, and I think when we talked about this case initially, we said, well, obviously we'd like to see the data, but it sounds like something that could happen.
Yeah.
Given what's been discovered recently about the persistence of the virus in semen, but now looking at the data, I had a lot of problems with this. So first of all, the patient S, the man, he enters he's estimated onset of disease September 9th. He goes to the hospital, gets admitted to the hospital September 23rd.
Mm-hmm.
September 28th, which is not shown on the timeline, they did an RT-PCR blood test on him, and that yielded ambiguous results.
Mm-hmm.
They tested again on the 29th with the same sample and got a negative result.
Mm-hmm.
So he's not Ebola virus positive yet. Then they subsequently tested on October 3rd, and they're not sure whether that was a separate sample or not. It was also negative.
Right.
And so he was discharged. So the problem starts here, where he's presumed to have had Ebola virus, but he never tests truly positive for it.
Did he get sick?
Yeah, why did he go to the hospital?
He got sick, and with diseases consistent with Ebola virus, and he went to the hospital, and they admitted him because they figured he probably had it.
Right.
And he had he had risk factors. Several members of his family had it.
Yeah, his former wife died. Yeah,
his former wife, who he was estranged from at that time. His older brother died of it.
Right.
Was confirmed positive.
Right.
And then then the S, the survivor, is thought to have had symptoms beginning September 9th, but we never actually see him come up positive.
Not before the semen, anyway.
Not before the semen sample. So, and then we get all the way to March 7th.
Right.
So we're 175 days or 155 days after he's tested negative for Ebolavirus. He and P, not to be confused with P, who we talked about in a different paper, had sex. And then P, a week later,
she gets sick.
Got sick.
All right. March 14th. Okay. March 19th, she goes into the Ebola treatment unit.
March 19th, she goes to the Ebola treatment unit. 20th, her blood sample tests positive. Then the 27th, they take the first semen sample from S.
It's positive.
And that comes out Ebolavirus positive, and P dies.
Right.
And then in terms of sequencing, that's the only sample they have from S.
Yes.
Is that semen sample.
Right.
Because the only other positive sample was the first time he went to the hospital, and they don't mention sequencing that one.
And that one was not actually positive; it was ambiguous.
Okay.
So S and P have sex on the 7th. S first positive test for Ebola virus of any kind, definitive positive, is 20 days later.
Mm-hmm. And then again, it's repeated, right?
So I'm kind of wondering. First of all, if this was sexually transmitted, who gave it to who?
Exactly. Let's look at his semen was positive once.
His semen was positive once.
And then two subsequent ones were negative.
And then two subsequent ones are negative. His blood sample is never positive and was not positive on his first admission for presumed Ebola virus disease.
Mm-hmm.
So I'm wondering if he ever really had it, or if this is an artifact, and that sample that tests positive, we're going to get to the sequencing in this paper, but they do sequence analysis on P's virus and S's virus, and they can't actually get sequence for S first. They have to do some additional manipulation of the sample and isolate the RNA and amplify the RNA through an additional PCR step in order to get any kind of sequence, and the sequence they get out of it is really crummy.
Is there any test for antibodies there? I might have missed that statement, but
no.
They just looked for the virus and not whether he'd been exposed or not to the virus.
Right.
No. Well, if you have
that's a good question, Dickson.
If you have Ebola virus in the blood, right, that is you're assumed to be exposed to it.
Understood, but maybe he already had been exposed to it a long time ago and harbored a latent form of this virus, as we're now seeing.
Or he could have antibodies which have modulated the...
So the antibody...
they didn't do that, no.
Yeah, they should have.
So the sequencing shows that his virus, all right, and the woman who died in his virus, is that correct?
Yes.
Differ only at one position.
Right.
And 15,808 nucleotides. They conclude these results are consistent with transmission from the survivor to the patient. But why would they be, with one base difference, how could you tell, right?
I don't.
Well, there are also a number of nucleotides different from other viruses that are circulating around. Those two viruses differ from each other.
But these two are the same, so there's no way you can tell the directionality of transmission, as Alan said.
Nor can you tell
the directionality. Okay, right.
Nor can you tell that it's not a contaminant.
So, Alan, it's possible that she gave it to him, and it resided for, 20 days in his testes, because the 7th to the 27th when he tested positive, and then it cleared.
Right.
And that was the end of it.
In which case, in him, it would have been an asymptomatic infection.
That's right.
Now, the next paper is really going to address this in a way, but in this case, it's just not clear that he gave it to her. That's right.
It's not clear that we have. Well, there's so.
But the thing is, Alan. The paper said epidemiologically there was no indication of where she got it from, which doesn't mean that it had to be him.
Right. I don't. I mean, I'm not going to speculate on where she got it or where he got it. He has very clear risk factors. But what I'm skeptical about is that this idea that anybody got it through sexual transmission, because the sequence data are really, I don't find them all that persuasive. I mean, yes, they've got the virus. It only differs by one base between him and her, and those two sequences are distinct enough from all the other stuff that's circulating in that whole region that you can say that they cleanly isolate from the rest of the tree. But what I have a problem with is they haven't. You would see exactly the same thing if you had inadvertently contaminated the sample. Right? We know this from the XMRV story, and given that they're doing this additional manipulation to the sample, in which they extensively modified the RNA isolation kit and amplified the signal further and further in order to get any signal out of his semen sample. It seems to me the risk of a cross-contamination there is quite high, and you would see exactly this sort of result. You'd get an identical or very nearly identical sequence because it's the same virus.
Sure. Now, if it's contamination, then you can't conclude anything. But let's assume that these were bona fide isolates.
If they're bona fide isolates, then I think we don't know the direction of the transfer.
And since they had sex on the 7th and she got sick 7 days later, that is within. She could have been incubating virus already on the 7th.
Exactly.
Right. So that would work. But this paper is very strong at concluding that he gave it to her.
Yes.
Direct transmission. They say the nearly identical genomes place the survivor and the patient in the same transmission chain. That's correct. And case tracing confirms contact by vaginal intercourse. So they're not making any conclusions there. They're just saying there was contact, but they don't know the directionality. Yep. Okay.
Yeah. So the argument they're making, I see why they're making that argument, and certainly their evidence doesn't exclude it. But I didn't find it.
It's not compelling.
Really persuasive. I think it's plenty to advise people who've had Ebolavirus that they should use condoms or abstain from sex, and that this is a potential risk.
Condoms anyway.
Yeah, you should be doing that anyway. But
not.
This is an argument that this is a possible means of transmission, but I do not think that this paper proves that happened. I'm open to that being the case, but I would agree. Skeptical of the data that they have on it so far.
Now there was one other case that they refer to in this of apparent sexual transmission of Marburg virus. Is that correct?
Apparently that was confirmed, and they got virus out of that case. So that is
which I think we talked about.
Yeah.
So if you're 11,000 strong out there that have survived this virus infection, and if this is a common route of transmission after surviving...
I wouldn't say it's common, Dickson.
Well...
It's rare, and they say that in both of these papers.
How rare? 1 in 11,000?
Yeah.
It is clearly very rare, because if it wasn't, then this would already be the new HIV.
Well, that's exactly what I was going to say next. That's exactly
this thing would already be everywhere.
Exactly.
And instead, by treating it as a strictly bloodborne disease and implementing the protocols to contain it, it's been almost successfully contained, and we've got chains of transmission that make perfect sense with it as a bloodborne disease. I think if it is sexually transmitted, which is possible but not yet proven in my mind, then that is going to be a very unusual path that it takes.
So recommending the use of condoms could easily be countered by the people that don't want to do that by saying, this is so rare it'll never happen to me.
Of course. And then we'll have more epi data for whether it...
Yes, exactly. And then we'll get definitive data.
That's right.
But it's we don't know if there's infectious virus. No one has got that from this case.
Right.
They say because we got complete genomes, it means the full genome is there, but it doesn't mean it's in one piece. I can imagine a scenario where the virus gets there and it just slowly decays.
Yep.
And you got pieces of RNA, and that's what we're picking up for a long period of time. So, until we have virus, just this one case for Ebola, it's not compelling, I think. Now, the next paper addresses this in a broader way. It's called Ebola RNA Persistence in Semen of Ebolavirus Disease Survivors, a Preliminary Report. And I should say that the first paper, the title is Molecular Evidence of Sexual Transmission, which is okay because they don't imply any directionality.
Right.
Lots of authors. The last author is Gustavo Palacios, who used to work here with Ian Lipkin, from a variety of people. Okay, so the second paper, they take 93 male survivors of Ebolavirus disease in Sierra Leone, different times after recovery, and they get semen samples, and they look for Ebolavirus RNA by RT-PCR. Okay?
Yep.
49% had positive RT-PCR.
Interesting.
46 of 93. And when might you ask, was this after infection? Okay. All 9 men who had a specimen obtained 2 to 3 months after the onset of Ebolavirus disease, the RNA was in all of them. It was in the semen of 26 out of 40, 65%, who had a specimen obtained 4 to 6 months after onset, and in the semen of 11 of 43, or 26%, who had an specimen taken 7 to 9 months after onset. One participant had a specimen taken 10 months after onset, indeterminate results. All right, so they have a lovely graph which basically shows these results. As you go out, the closer you are to onset, the more detection, then it goes down, and the no detection goes up. So the further you get away from the original onset of disease, less and less RNA is detected. So my interpretation is that
this is a decaying virus?
The virus is, during the infection, it's viremic. It's getting into lots of tissues. It gets into perhaps the testes. We don't know for sure. And it sits there. I don't know if it even replicates, and it slowly decays because there's nothing countering it or destroying it. And then you detect it in lower and lower amounts. The real question is, when are you infectious, if at all? Because maybe you're infectious during the height of disease when you have the most virus, but you're too sick to have sex, right? So that's the thing. There's clearly persistence of RNA, but whether it's infectious virus and whether you transmit it, we just don't know.
So could you set up a cell culture system to prove that?
Well, they try to recover virus from these individuals. The guy in the previous study, they put it into cell culture, and they can't get any infectious virus out. Okay. I don't know if they did it with these samples. This was just a PCR study, but they do have the samples. I'm sure they're going to try.
Yeah.
They state clearly that they're just looking at RNA, that they don't know if there's infectious virus there.
Oh yeah, here it says on the last page, virus isolation assays are underway.
Right.
So, it's there. RNA is there. What the risk is unknown.
Right.
But in the meantime, it's not a bad idea to use a condom for a certain period after you're infected. And what CDC recommends...
It's not a bad idea anyway.
It's not a bad idea. It's
just a great idea, actually.
Well, it depends, Dickson.
Well, if you don't want to have kids, that's a great idea.
What if you're 80 years old and you've been married to the same person for, 50 years? Do you need to use condoms? Come on, Dickson.
Well, it depends on what you just got over.
Well, there are lots of conditions. Fine, I get it, but don't make a blanket statement. We'll get letters.
A blanket statement. This is an undercover statement.
To me, one of the most significant things here is that it's detectable out to 9 months. That's a long time.
That's a long time. That's right.
RNA is detectable.
Yeah. How does that compare to other acute infections like influenza or smallpox? Of course, that's that.
Dickson, how about herpes? How does that correlate with
herpes is a latent infection. But I mean, for those other infections, how long does the RNA persist?
Well, influenza, not 9 months, but I can tell you poliovirus will persist 20 years.
Wow.
In a person who doesn't make antibodies.
Wow.
I'm wondering if some of these individuals have some subtle immune defect of some sort that...
Well, it is said, I'm no immunologist, but it is said that the testes are another immunologically privileged site.
But I'm starting to wonder about this. We always thought the CNS is a privileged site, but now as more and more people look, they're finding immune responses, right?
Well, privileged, I think we're just reinterpreting the word privileged.
Yes.
It's not black and white, right?
There's a very, very active set of immune cells in the brain. We now know that.
Yep.
So it's not immunologically deserted.
Exactly.
But it is clearly immunologically privileged in the sense that stuff that's going on the body side of the blood-brain barrier doesn't get there.
Right.
And so there's a certain privilege to that compartment that things may not get cleared as effectively. Mm-hmm. And so, they and same with the inside of the eye, the vitreous humor, and same probably with the testes, that there's not the same level of clearance that you might have in the bloodstream.
What's the current recommendation for condom use after Ebola virus recovery? How many? 3 months by CDC?
I think
6
it
months.
was 3 months, but they bumped it up in light of some of these data. So is it 6?
Although no virus, blah, blah, blah. WHO and CDC has revised their guidelines regarding the length of time, and it doesn't say what it is here, 12 and 13. Advice on, let's take a look. I'll just click on this. Wow, the magic of the HTTP protocol.
Yes.
Here we go. Risk of exposure, diagnosis, prevention. Gee, what? Transmission?
3 months.
3 months.
Offered semen testing for 3 months. And for those who test positive every month thereafter until it tests negative twice by RT-PCR.
Wow.
If you have not been tested, continue to practice safe sex for at least 6 months.
6 months.
This is the WHO.
Okay. So I would say that the nurse story and these don't really give us a sense. Well, in her case, we don't know if... I guess there was virus in the CSF, right?
Right, but we don't know if that's...
there was something in the CSF.
All right.
Right.
So certainly virus RNA can persist in certain places after it's gone from the blood, right?
Right.
The exact risk of that is not clear yet.
Right.
It certainly isn't like HIV, right, which is sexually transmitted.
Clearly.
Okay. Anything else, Dickson?
Well, one of the things
a couple of good recipes for barbecue, but we can save that for some other time.
One of the things that I keep thinking about is that there are sequelae of paramyxovirus infections. These are related viruses. I'm thinking of SSPE, subacute sclerosing panencephalitis in the case of measles.
Right.
And that is measles virus genome that persists in nervous tissue for, years afterwards, but it's not actually virus, if I understand it correctly, and it's not clear how this causes pathology. So this question of whether we're talking about infectious virus or whether we're just talking about RNA is an important question that I'm sure we'll hear more about as time goes on. But it'll be interesting to find out whether these actually situations actually represent virus infections that are persistent, or whether it's some other sequelae of the initial virus infection that genetically is a little different.
Yep.
And the other issue is that it's clear from just these reports, we now have at least 11,000, and that's probably not 11,000, on the order of over 20,000, over 15,000 individuals who have recovered from the disease. Right. And there's going to be more sequelae that we're going to learn about as time goes on. This is going to be an issue.
Yeah.
Yes.
Okay. It's enough. Do a few emails.
Yep.
There's your Ebola update, gang.
Yep.
That's right.
That's right. Your Ebola fix for the month.
All right. Now, a fellow on Twitter today, his name is Baker Virus Maker, said, I seem to recall a TWiV podcast pick of the week that involved an antibody rating website, but I can't find it. Help. Does anybody... I couldn't find this either.
This sounds familiar.
Yeah, someone sent in a site where you could rate your antibodies, right?
Yes.
And we talked about it.
I thought it was even linked in an episode.
It wasn't anybody's pick?
Yeah, I looked in the pics and I couldn't find anything with antibody in the title.
Somebody wrote in. Somebody wrote in with this.
And it was not a pic, so it would be in letters, right?
Maybe not. It would probably be in letters.
All right, I have to go and look through it this weekend then. I thought you guys would.
Whoever it is probably listening and will send us an update.
All right.
I hope.
Yes.
Well, whoever's listening and knows what we're talking about, because if you just search for antibody rating, you get tons of sites.
Oh my gosh, yes.
But there's one that was recommended that was good. All right, Alan, can you take the next one, Bill?
Bill writes, many have tried to apply the lessons of the immune system to treating computer viruses. Alas, the comparison is not that useful. They share the math, but with wildly different contact rates. But a number of TWiVs ago, you folks mentioned superinfection suppression, wherein a virus prevents interruption by new infectors. Hackers do this routinely, and here's a recent Slashdot article about the practice. And it provides a link. I love my weekly dose of hard science from you guys, even if it isn't exactly in my field. Cool.
I would guess, though, that a lot of digital viruses hit different things so that they wouldn't necessarily prevent another one from coming in, right?
Right.
Although if it erases your hard drive, nothing else is going to come in, that's for sure.
That's right.
So this is great. This is Shifu is a banking Trojan that's currently attacking 14 Japanese banks. Once it has infected a victim's machine, it will install a special module that keeps other banking-related Trojans at bay.
Yes.
It really is super infection exclusion. I love it.
Wow.
Oh, Rich Condit. Can you take the next one?
This is a different Bill, I hope.
Yeah,
it is
Bill writes, decline in academia. This blog speaks to it. And this is a blog by an individual who was a postdoc, I believe, in neuroscience or something like that at Duke. It's been a long time since I've read this. And he blogs about having resigned his position because he's sick of the rat race. And he's in particular sick of the publishing rat race, where people are scrambling to get into vanity journals, and it's, in his opinion, not based solely on the science, but as much on who you are and how much you whine. So it's an interesting blog.
That's the way that the field is. It's tough. It's not for everybody. But what he's really doing, he has a podcast called Neuro.tv, and that's what he's doing.
Yeah. Good for him.
It's got 16 episodes. Monthly conversation between neuroscientists, psychologists, and philosophers who try and understand the brain and the mind. I will have to check it out.
Yeah. Rich, sounds like something you're trying to do right now.
Dickson, did you quit because you were fed up with the rat race?
Absolutely. No, I didn't quit. I just did something else.
Yeah, I didn't think you were fed up with the rat race.
No, I'm not fed up with it. I enjoyed the rat race, actually.
He loves racing rats.
Look, the moment you get tenure, it's not a race anymore. You won.
I think that the pleasure of doing science is well worth it, whatever crap you have to put up with.
No, I agree.
Discovery is just amazing, right?
I totally agree.
Rich, you want to take the next one, too, since that was rather small?
Sure. Judy writes, thanks for all you do. Use this on TWiV or TWIP as you see fit. Judy is a retired science teacher. She says it is sunny and 75 degrees Fahrenheit, 24 degrees Celsius in San Diego, light breeze, 52% humidity, just another day in paradise.
She's right. It's always like that.
Not always. I was there and it was raining.
Really? That's very unusual.
That's where Judy's from. I was out there.
Very unusual, especially in California.
Because when I went there.
So she links to an article about, we've actually talked about this before, about immunizing against rabies in the wild. It's in particular an article about doing this in Switzerland. The article's titled "Switzerland's Kind of Gross, Incredibly Effective Anti-Rabies Weapon." It's actually dated 2015, but this has been going on now for decades. And just to summarize, in numerous European nations and in North America, where rabies is the primary reservoir is wild animals of one sort or another, raccoons, foxes, and skunks. One of the programs to get rid of it is to distribute in a baited form a recombinant pox virus that expresses the rabies virus glycoprotein, so that animals eat this bait and they're immunized against rabies. When in Switzerland, when they first started this, they distributed it. They're dropping it, I think, out of airplanes. That's one of the ways they do it, in chicken heads.
Right.
So that's the kind of gross part. But subsequently, they figured out other ways to bait this. It's
now they use something that's kind of like a doggy biscuit.
Yeah, it's enormously effective. And to me, one of the interesting things about this is that this is we worry about. Well, some people worry about distributing or using recombinant virus vectored vaccines, and this is a situation where this particular vector has been just, basically, dropped out of airplanes all over the place, millions of doses all over North America and Europe, and it's done a fantastic job, actually, of knocking back rabies.
Sure.
And we've reported on just a couple of cases where there's been a problem, which is one that I remember was some woman, her dog retrieved one of these things and brought it back to her, and she had some cuts on her hands and handled this bait and got some virus in her hands. She was immune suppressed. All of the situations where there's been a problem, been immune suppressed individuals.
And
At any rate,
she had been picking blueberries, so her hands were cut up.
Right. So this is, at any rate, this is a nice summary of the whole thing that was sent in.
So the U.S. Army drops this bait all the way across the U.S. border between Mexico and the United States because of the migration of coyotes primarily, and I've read articles about this. But I think Donald Trump has a new way of actually doing this without the use of rabies vaccine.
Yes.
Hey, Dickson, are you up for an email?
This is a long one, and I'll give it my best. Ted writes, TWiV 349, one ring to vaccinate them all. Again, elicited responses and references to contemporary anti-vaccinators. 4 years after Edward Jenner's death in 1823, the Boston Medical Intelligencer, one of two predecessor journals to the New England Journal of Medicine, printed a number of articles concerning smallpox, inoculation, vaccination, and a review of a just published book, The Life of Edward Jenner, MD, LLD, FRS, Physician Extraordinary to the King, with illustrations of his doctrines and selections from his correspondence by John Barron, MD, FRS, which reflect wisdom undiminished by the passage of 188 years. The April 17, 1827 issue reported that although the Association of Boston Physicians did not approve a general vaccination for smallpox at that time, they recommended that when vaccination was required, the poor could apply to the mayor for a certificate for free vaccination. They also proposed that the Boston City Council apply to the legislature for an act giving power for the municipal authority or school committee of any town or city in the Commonwealth to make a condition of admission to the public schools that children shall exhibit satisfactory evidence of having undergone the cowpox. Emphasis my own. Additionally, should the legislature pass such a law, they proposed that a general vaccination institute be established for the preservation and dissemination of the vaccine matter. Interestingly, the Boston Common Council subsequently approved this proposal, commenting, "It is the first adequate and effectual measure of security we have ever enacted against the introduction of smallpox since Edward Jenner announced the vaccine preventative," emphasis my own, though they left adoption and execution of the proposal to the Commonwealth. Also in April 17th issue, there is this report from the Philadelphia Museum. In other countries of Europe, general vaccination is ordered by government. No one who's had the cowpox or smallpox can be confirmed, put to school, apprenticed, or married. Smallpox inoculation prohibited. If it appears in any house, this house is put under quarantine. Such, I'm having problems with such means, oh, come on, stop it. I'm trying to get out of this now.
Yeah, is your
yeah,
iPad doing
some
funny
pop-ups. That's okay. By such means, the mortality of smallpox of 1818 has been prodigiously lessened. Emphasis my own. On December 19, 1827, there appeared this reprint from the New Haven, Connecticut Herald newspaper. The case of smallpox occurred in the city last week, which terminated fatally on Sunday. The young lady who was the subject of it had been keeping school, but how the disease was communicated is not known. We are informed that another case exists on York Street. Yale New Haven Hospital is presently located at 20 York Street. We mention these facts as well for the purpose of cautioning others as to express our astonishment that any person in this community should have arrived to years of discretion without having been the subject of vaccination, the only sure remedy for this dreadful malady. The ease and facility of vaccination leave no ground for excuse, and parents and guardians must lay to themselves the neglect of an important personal and public duty and bear the charge of bringing evil on the city. Emphasis my own. The October 9, 1827 issue, containing a review of Jenner's biography by his lifelong friend John Barron of Gloucester, Massachusetts, in part states, to inoculation, vaccination followed, which is fairly characterized to be one of the greatest improvements ever made in the practice of medicine. During the last 16 years, the practice of vaccination has been fixed on a permanent basis. Its feeble enemies, feeble even in mischief, have sunk into oblivion, and though it has not, like the test of a literary reputation, survived its century, little doubt can be entertained of its eternal success. Again, emphasis my own. The words of George Santayana are indeed true. Those who cannot remember the past are condemned to repeat it. Again, many thanks for your wonderful podcasts, Ted.
And Ted has written before. He's a professor.
I like it.
Down there in Florida.
I like it.
Of oral and maxillofacial surgery.
Cool.
And, this it's well known that there were anti-vaxxers in the Jenner era. There's a wonderful cartoon you can find at the Wikipedia entry for vaccine controversies entitled The Cowpox, or the Wonderful Effects of the New Inoculation.
Yeah.
People growing cows from their Jenner sites. Yep, that'll happen.
Right.
Thank you, Doctor.
Well, I had to have proof of smallpox vaccination to get into kindergarten.
Oh yeah,
remember that? Yep.
Interesting.
I don't know if I was required to have proof of it, but I got it.
I don't have my scar. I wonder what happened to it. I know
they can disappear over time. It's hard to find mine.
Yeah. What's interesting is to see all these ancient movies of historical importance back in the the Crusades, and he bears his chest to fight with the evil villains, and he's got a smallpox scar on his shoulder.
Yep. All right, let's do some picks.
Oh, good.
Alan, what do you have?
I have. This was actually sent to me by a source for a story that I'm working on. This is not directly related to the story, but kind of tangentially. It's a little animated short film. It's about 20 minutes. The title of the film is Sticky. Uh-huh. And this is the story of the Lord Howe Island stick insect, also known as the phasmid, which... Lord Howe Island is a little fleck of rock between Australia and New Zealand. It's administered by Australia. And of course, it had its own island ecosystem with all its own indigenous species of all sorts, including this amazing little stick insect. It's actually big stick insect. It's about the size of your hand.
Right.
That fed on the vegetation there. And... that shortly after the introduction of rats, that insect vanished and was believed to be extinct. But it was, for a short time in the 19th century, it was absolutely fascinated Europeans. There are collections of it all over the museums, and wow, this is a neat insect, and then it was gone. And then there's a little coda that's a very common story. But there's a coda to this one, which is that in 2001, some researchers located on it went to confirm that this thing had not actually been placed anyplace else. and they went to an adjacent rock that's sticking out of the ocean about a half dozen miles away. And people had theorized, well, maybe there's a population of it over there. And so they climbed up this rock to confirm that it was not, in fact, there, and they found a population of it existing on a single bush on this rock.
Kidding.
Wow.
And they then started a captive breeding program.
Right.
So it's this animated film is just absolutely gorgeously done, and I've watched it twice. I'm probably going to watch it again.
Oh, that's
it's just really nicely done and lays this whole story out. And the intro of it, before the narration starts, there are no words in it, but it just graphically represents the whole first part of the story, and it's just really, really poignant.
So why don't we use the rat virus to wipe out the rats on the other island and then reintroduce the stick
They are
insects?
now talking about, yes, watch the video. Yes.
Oh, good. Because we have to connect the circles here, don't we?
Yes, rats play a prominent role here.
When I saw phasmid, I thought of phagemid.
Yes.
Yeah, me too.
Phasmids, it's a term used in nematology.
I saw.
There are nerve endings that actually come out the front end of these parasites that they use to detect their environment with. And also, free-living nematodes have them also. Amphids are in the back and phasmids are in the front.
And can you set any of them on stun?
You get that, Dickson?
I did.
Just have to make sure. I want to make sure you're
So,
up.
no, you keep accusing me of antediluvian activities. I watch all the local movies. Come on. I'm with it, okay? I'm with it.
Rich Condit, what do you have?
So I went just a couple of nights ago and saw the new Steve Jobs movie.
Mm-hmm.
Oh, yeah.
And that starts out with a little clip that I was able to find on YouTube of an interview with Arthur C. Clarke from 1974, where he's in some room with a, huge IBM computer of something or other, and he's talking to the interviewer and his son, and predicts quite clearly that someday everybody's going to have either a terminal or a PC in their house, and they're going to be able to communicate with everybody else, and it's going to be wonderful because, you can telework and all of this kind of stuff. So he basically predicts the blossoming of the personal computer and the internet.
And of course,
So I thought that was pretty cool.
He
There's
wrote a lot
little
about
YouTube
HAL,
video.
so that's obviously true.
And I didn't want to steal your thunder, but I put another link underneath that for you, Rich.
Well, can you trump a pick? This is a new thing.
As soon as I saw this, I watched the video. I said, "Oh yeah, I've got one."
Yeah. So Alan has here that there's Vannevar Bush apparently predicted the web and Google Glass even 30 years before this, and Alan has a link to a magazine article. I haven't read it yet, but
Bush has a very,
cool.
very famous essay called "As We May Think," which may have inspired Arthur C. Clarke's thinking on this matter.
Could be, yeah. Okay.
We all stand on the shoulders of giants, don't we?
That's right.
Here.
Don't we, Dickson?
We sure do.
Dickson, what do you have?
Well, they just released the winners of the Nikon Small World Contest yesterday, so I thought this would be an appropriate time to put that in there.
Yeah, Dickson, it's
I don't
beautiful.
see any viruses there, though. They should have a couple of crystallized capsids or something.
They're not. You can't do color unless you colorize them.
Well, these are all colorized.
They are colorized, yes, of course. Many of them are. Are all of them colorized?
All of them are in color, but I think, they've got false
I guess you could
colors.
colorize a virus, sure.
Sure could.
Just need an electron
The
microscope.
first one, the winner, though, the eye of a bee colored with pollen grains.
Oh, look at that.
It's just so beautiful. And look at the crispness of the photograph and everything. It's
That's
just
neat.
It's fantastic.
And, Dickson, it seems like you just did this for last year. They come up quickly, or
They
are
come
the
up
years
once
just
a year,
passing?
actually.
Are the years passing quickly?
It's like the Nobel Prizes.
There are a couple of these. The Nikon Small World is the big one, but there are a couple of other scientific imaging.
That's true. And they've expanded their categories for this, too. Now they have videos, and they have lots of other categories as well.
Well, Dickson, the second place winner is the mouse colon colonized with the human microbiota.
Yeah. Isn't that
That's
cool?
pretty nice.
That is.
The third place is really... Oh, I like the third place a lot.
Fantastic.
Intake of a humped bladderwort.
What the heck is a humped bladderwort.
A freshwater carnivorous plant.
I never heard of it.
That's beautiful. I like that the best, actually.
It's fantastic. Do you?
I
That's
do.
neat.
And I like this sixth place one, too. A spore capsule of a moss.
Yeah. It's fun to play a game with this. Hide the caption and see whether you
what
can
it
tell
is.
if it's an animal or a plant. It's hard to do.
The starfish is cool.
How do you get a confocal microscope picture of a giant animal?
Well, you can do that. No problem. No. Just back off on the magnification.
Wow.
That's beautiful, yeah.
They're all
Science
gorgeous.
art, huh?
That's the point. What they're looking at here is patterns of beauty, but these are all, that's the way nature behaves. And so we're in awe of it every time we see these. We just go, oh my God, look at how beautiful that is.
Transgenic xenopus, tadpole head expressing green neuron.
They're fantastic.
Oh my gosh, it's beautiful.
Yeah.
But the tech behind this too is the science is pretty complicated.
Yeah, absolutely.
Sure.
You just can't
Absolutely.
take a piece of dirt and take a picture of it anymore. All right,
Well, yes, you can.
you can do it, but you're not going to win the
That's also
contest,
true.
right? So my pick is a blog post by Pat Schloss, who is a microbiologist out at the University of Michigan. Of course, I met him last week, and we have talked about two of his papers over on TWiV. He studies the microbiome. He's also a computer programmer. He writes the software that he uses. He's well trained in that, and he wrote a program called Mother, which is widely used in microbiome studies. But anyway, last week there was an announcement in Science magazine by U.S. scientists calling for an interdisciplinary unified microbiome initiative,
Aha.
the UMI. And then he says, not to be outdone, Nature published a similar piece indicating that each country would have its own UMI, and these would be assembled under an international microbiome initiative. And I wonder, will you use Mother to assemble that? Pat, that's a joke, because it's one of the things I think Mother can do is to assemble sequences. Anyway, so initially he had a negative reaction to this, and he said, we have a global effort over on Twitter. It's called The Scientific Method, which is great. And then he backed off a little, and he talks about his thoughts. And the last part is the best, where he has some bullet points of what he would do differently or in this announcement. And one of them is a return to boutique science. Having participated in several large initiatives, I'm ready for more small science. And I think that is wonderful, because big science can be very wasteful, and it's not necessarily more productive, right?
Yeah, there have been a couple of articles recently about the efficiency versus lab size that basically show that the larger labs are not as efficient as the smaller labs.
Right. And
Look at, I'm sorry.
It's okay, Dickson.
I wanted to go back to my pick, because as you start to look through those images and see how they were taken, the variety of high-resolution microscopy has gone bananas. There's a whole department of high-resolution imaging at the University of Nottingham, for instance.
Dickson, it's called super resolution microscopy now. Yeah, it's a combination of optics and also software.
It's fantastic.
Yes, they do really amazing things. You should actually go talk to someone doing it, Dickson, to find out how really amazing it is.
Yossi is right down the hall.
Yeah, we're not an amateur.
Oh my God, I'm going to tell him you said that.
It's an amateur mic. Well, he's always out there making noise while we're recording, so he needs to
he
needs to quiet down.
Gotcha.
You see, I'm really cranky right there, Dickson. Anyway, this is a wonderful post by Pat. Check it out. His blog, he's got a blog too, the Mother blog. Check that out as well.
Get him on board by saying UMI actually stands for the University of Michigan Initiative.
That's very funny. While we were at this ceremony, this milestone ceremony, the president of the University of Michigan actually came for 10 minutes and gave a talk. And he happens to be an immunologist by the name of Mark Schlissel, whom I know because we looked at him as chair many years ago. And he said the next day I'm going to make an announcement. And he made a $150 million initiative the next day to improve science. And,
wow.
It's much more than that. But he's really supportive of science at Michigan. Mm-hmm. And we need this.
Absolutely.
We could use this at Columbia.
Well, we just raised $2.5 billion.
You're mistaking Columbia for a university, not a real estate holdings company, which is what it is.
It's Dickson. It's Manhattanville, the new campus, which is very expensive, is not just for science. I mean, science is a component of it. But I don't need new buildings. I need supportive faculty.
I hear you.
To do their work.
I hear you.
You know, we don't get enough of that here. I appreciate that Columbia lets me blog and podcast, but we need to do more. All right, we have a couple of listener picks. One is, they're both via Twitter. One is from, did I put the link in it?
I don't see it.
Ooh,
I've got the one from
here it is.
Ian.
So, Awkward Skeptic retweeted an Ian Mackay post. A 16-year-old wins the Google Science Fair with a way to detect Ebolavirus. Did we mention this before?
This
sounds familiar, doesn't it?
I don't know that we have mentioned it.
All right, so that's a Google Science Fair winner, and you should check that out. And that's over on Ian Mackay's blog. Well, it's probably a Scoop It thing. Yeah, it's a curated site. Emerging Viruses, Virus Discovery, and Virus Characterization, curated by Ian Mackay. And so check that out as our listener pick of the week. And that will do it for 1 degree past 360 of TWiV. 361. You can find it at iTunes and TWiV.tv. And did you hear? Google is now going to be putting podcasts in its Google Play site.
Ooh, cool.
So iTunes does podcasts and music, right?
Yep.
Google Play sells music. Yep. Now they're going to put podcasts in it. And that's good for us because we could get more listeners. So I have applied for all of our podcasts to be admitted because you have to be admitted. But I'm sure since we're academically rigorous, we should get in, right? Or maybe that's not enough. Who knows? Anyway, I don't know when that's happening, but check it out. If you're an Android person and, you want to hear our podcast.
Even check it out if you're an Android.
See Androids Fighting. I bet you don't know what line that movie is from. And if anyone has listened this far, you tell me.
It's got to be Star Wars.
Nope. TWiV.tv, iTunes, and if you have questions or comments, TWiV at TWiV.tv. Dickson Despommier, thank you for joining us today.
You're so welcome.
Appreciate your contributions to the elucidation of viruses.
I try.
Rich Condit.
Lord knows I try.
Lord knows I try. Rich Condit is in Tampa, Florida today. He is an emeritus professor at the University of Florida at Gainesville, and I really appreciate that you have connected on your little during your little sojourn there, Rich.
Well, I wouldn't miss it for the world. Anytime I can make it, I will do so.
Thank you very much.
So it's always a good time.
And you'll be home next week because we have...
all right, we got an early recording.
An early recording. Alan Dove can be found at turbidplaque.com. You can find him also on Twitter as Alan Dove. Thank you, Alan.
It's always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.